Okay, good afternoon, viewers at home. My name is Olatunde Michael. I am a lecturer under Omega Bioedutainment Hub, where we are passionate about bringing biology courses nearer to the people, larger populace. Don't forget to subscribe, hit the notification button so that you can be part of us anytime we come online for educational series. This morning, we are coming up on a very simple, the beginning of microbiology. So, the beginning of your journey as a microbiologist. In the past few contact hours, I've been able to elucidate so many things, which include history of microbiology, that is the beginning and the origination of microbiology, where we basically made mention of some specific microbiologists and adapt with their contribution in microbiology generally. And we look at some lines, timelines, and milestones in microbiology. This morning, without wasting our time, we are going to further our introduction to general microbiology. And if we could do any course in microbiology without knowing details or part of what we want to discuss today, honestly speaking, we might not have a very smooth journey in the area of microbiology. Specifically, today, this is a general microbiology. General microbiology. Okay. So I told us that we've already had introductory classes on history in the past. In other courses, that is previously, we have been able to look at the scope, branches of microbiology. Okay. After that, we also look at the history and milestones, where I give us lots of acronyms or the shortest formula that we can use to understanding microbiology. So milestones in microbiology. Okay. Now we also look at we also dedicated one of our previous contacts to understanding the branches of microbiology, and with specific focus on bacteriology. Okay, bacteriology. So whereby I gave us a typical structure of a bacteria, bacterial cell. Okay. Then we look at the functional parts. We look at the structure and functional parts of bacteria. So, and we were able to start picking at a bacteria where I draw something like this for you. Okay. And with some kind of this. So we started with a cell wall. I told you the components of cell wall. We look at a flagella. Okay. We look at pili, or what's called pilus. Okay. Then we look at the genetic material, that's our DNA. Then we also look at what plasmids are and the function part of a plasmid. Okay. So also I also mentioned something about ribosomes. Then we also look at inclusion bodies. Okay. So apart from that, we also look at we are able to look at classification or classification of of microorganisms. Classification of microorganisms. That was where we started, and that was why we talked about bacteria, viruses, um, fungi. And I told us that the study of bacteria is called bacteriology. The study of viruses is called virology, whereas the study of fungi is called mycology. So we look at that. Then we also ended it with the general characteristics of bacteria. That is, what are those things that you will see so as to give you the clue of where to classify your bacteria into. And I remember that when I mentioned something about classification according to the cell wall, I said bacteria can be classified into gram positive and gram negative. I gave us example in each case. I gave you example of here as E. coli. Okay, that is gram. So, and this one I gave you example as a staphylococcus species. Okay. So I said the distinction between gram positive and gram negative is that this one has a thicker cell wall, whereas that of gram negative, their cell wall is thinner. Okay, and I also went ahead as telling us classification based on the sizes, the characteristics of bacteria based on their sizes, shapes, and I gave us different examples in each case. So that is a previous peep into the last lecture we had in this. Don't forget that this is the beginning of our journey in microbiology, and all these basic we're supposed to make sure that you understand this. If you do not have any good background on microbiology, your journey there might not be that easy. Okay, so we don't want to waste our time today. We are going to focus on our business of today, and the business of today, I said, about 3 to 4 videos is going to be dedicated for the next few days. The first thing is that we are going to focus on the basic techniques in microbiology. You have to be, what do we mean by aseptic condition or aseptic technique? We are going to focus on that. We are going to focus on what do we mean by microscopy. How do we carry out microscopy in microbiology? And staining, that is microscopy and staining. We are going to combine these two together. After that, we will now look at how can you collect samples, and by the time you collect samples, how can you handle it? Because if you happen to collect a wrong sample or a wrong sample, that will go a long way to be able to determine what your result is going to be. That is why collection is part of what you should know in microbiology. Some of us are going to start our project very very soon, and we will start collection. We will start, that is the beginning, start collection of uh, samples from that. Then apart from that, we could collect very good samples, but we may mishandle them. So we are going to look at all this as our focus in the next basic laboratory techniques. Then after that, we are going to look at I said sample collection and handling. After that, we go ahead to look at what can we do in especially when it comes to handling. Handling includes labeling. How can you label what are those parameters that should form your labeling? So then storage is another very very critical part of our technique that we're supposed to know in microbiology. So after that, we'll now look at storage. We can have a very good, excellent sample collection, or you could transport it very well, you could label it correctly, and if you are not so careful, you do not know the condition that you need to keep your sample. Honestly speaking, the integrity of your sample is going to be badly affected, and this is going to go a long way as what is going to determine the overall results. Then apart from that, the timely processing of your results is very very important. It's not going to be that you collect sample last month or you collect sample last year and you start processing it from next year. No, it is very very important. So after we must have been used to these basic laboratory techniques, we'll now look at how can we cultivate and culture. What are the cultural and cultivational techniques? How can you culture them? Don't forget that you are going to bring them from their normal ecosystem, from their normal location. You are going to now, you want to study them. If you want to study them, it's like when you want to domesticate your animals. So you take it from where you are going to buy them. Okay, after that, you now keep them. When you keep them, that's also going to have that. So we are going to move to the second part, which has to do with the cultivation and cultural techniques. So different types of culture media we are going to know. We are going to talk about culture methods. After that, we will now look at microorganisms and diseases. I remember in my previous classes with you, I just told you that microorganisms might not necessarily be referred to be our enemies, or they might not be referred to be so they are both our friends and also our enemies. I gave us examples of those ones that are our friends. These are the ones that are very very important for us to maintaining the the normal body temperature between the environment and our body. So these are organisms that live on our body, that we call them the normal flora. I told us a very simple analogy that day, that sometimes you need to get a thief within in order to be able to settle or to be able to fight against effectively the thieves without. So it's very very important like that. Then we will now look at their importance. What role do they play when it comes to causing diseases? So we are going to see some of these, and this is going to be the third aspect that is going to be devoted for microorganisms and diseases. So after that, the last one is we are going to now consider preservation techniques. We'll start picking all these techniques one after the other, and by the grace of God, we are going to see how we can navigate our knowledge, our basic knowledge in microbiology. How can we navigate them very, very efficiently and very, very effectively. So please, in the next just 2 minutes or yes, or 3 minutes thereabouts, I want to really give you time so that you'll be able to copy as a kind of a review of what we have done in the past before we'll be able to start what we are going to do for today. Thank you. Thank you for your attention. So, see you in the next 3 minutes. Thank you. Okay, I want to welcome you back to the second segment of Omega Bioedutainment, and our focus today is on basic laboratory techniques in microbiology. So, and that is going to be our focus now. For the next few minutes, we are going to be talking about that. So, basic laboratory techniques. Basic laboratory. Don't forget that your training as a microbiologist will go a long way, will actually determine your basic laboratory experience, will actually determine how good you will be in microbiology. Okay, microbiology. Yes. Um, what are these basic? What do we mean by basic? Basic means the required, the necessary, the foundation, the beginning, the origin, what the ingredients. So it's just like if you want to prepare a very good soup, whether you are going to get a very good soup, will what is going to determine it is the type, the integrity of the ingredients you are going to use. If you are going to make use of a very bad ingredient, definitely your soup can you can evenly start from the beginning and even guess how palatable your soup is going to be. So the same thing. So that is why we call it basic. What are those basic techniques that you as a microbiologist, as a scientist, that you must know? The first thing you need to know, I want you to learn this, to put this one down. We are going to consider what do we mean by aseptic technique. So the first thing that you need to know is aseptic technique. In all your handling, in all your dealings in microbiology, aseptic technique is very, very important. So what do we mean by aseptic technique? Aseptic technique means that you prevent, the act of preventing, or the act of eliminating or eradicating or minimize. So you can say the act of eliminating, minimize, reducing, eradicating what? Contamination from your samples. So you need to learn how, whenever you collect any samples in microbiology, how are you so sure? Because your decision is going to go a long way to tell about the integrity of your sample. So if you collect a very good sample, very good handling, very good excellent storage techniques and some other, and by the time you want to start processing your sample, you are not so sure whether what you are seeing as being grown or in the on your plate is a contaminant. So you need to make sure that you do not, contamination should not be coming from outside, it should not be coming from you, and that's why the first thing you need to know is to do what is called PPE. You have to be excellent in PPE. PPE means personal protective equipment. You need to have some basic equipment. It's not a matter of maybe you will just, what you will do is just you just go to the laboratory, go and do anything. You have to be in your PPE. We are going to see a lot of this. So that's why aseptic condition, if you want to define an aseptic condition, I want you to really take time to write this down. So aseptic techniques are a set of practices that prevent contamination by unwanted microorganisms. So the art of making sure that you do not introduce or you do not let contamination to get into your sample, all the set of techniques including cleaning the surfaces where you want to work, that is your bench, cleaning it with disinfectants, washing your hand in between handling samples, the use of personal protective equipment, and so on, and ability to wear nose mask. It's not that by the time you start handling, that is when somebody will begin to talk. So you cannot prevent contamination. That means it is not an aseptic. So aseptic from the word that is absence of what? Absence of contamination. Aseptic techniques are those techniques that you make use of when you are handling your samples. So, and we are going to list some of these by the time we go on. I want to basically talk about 4 or 5 practices that you as a microbiologist you need to know about. I've talked about personal protective mechanism or equipment, so such as wearing of laboratory coats, wearing of lab coats, the use of a face mask, use of face mask, hand gloves, and so on. So these are what contributes to our personal protective equipment. So then the second practice is to make sure that you have a very sterile environment. Very sterile environment. That is an environment that will be very difficult for life to grow apart from the life of your organism or the life of your microorganism. So sterile, you keep it away from contamination. So this includes example of what you do to ensure a sterile environment is flaming, the use of flame, hot air. Make sure the air around you is either too hot for contamination or for contaminant to be able to grow there, or you make it too cold. That is the use of maybe you have air condition, you are working where the environment is so cold to the level that it cannot support the growth of a contaminant. So sterile environment can be maintained with flaming. Then you can also use what is called in modern laboratory, we have what is called laminar flow of air around, so that laminar flow, that such as this is an equipment that will be filtering the air around you, filtering the air around you, so that no microorganism or no contaminants will be able to grow there. Okay, that is the first basic. The second, this is the first one, your basic personal protective equipment is very important. Okay, so and the second one is sterile environment. You have to maintain a very sterile environment. So e.g. by the use of flame, when you look at your inoculating loop, don't forget this is how inoculating loop looks like. So this place is always made of wooden. Okay, so just like this. So the air you make sure this place is red hot. By the time you want to pick your organism, it's red hot. So very hot. This is a source of this. Then the hair around you where you are working, I told you that it is either the hair or the surrounding the condition around that place is too hot or it is too low. If it is too hot, that means you are making use of flame around you. If it is too hot, that means the use of air condition or some other cooling equipment. Okay, that's why normally if you want to arrest the growth of your microorganism, you keep them in refrigerator. We are going to see some of these. Then the use of a laminar flow. I told you laminar flow. This is a modern equipment that work around you to make sure that the air around you is well filtered so as not to allow the growth of a microorganism. Okay, the third one is that you have to apply your sterilization rule. Is excellence. Sterilization of tools. All the tools that you are going to use, including media, must be well sterilized, and that's why we normally apply what is called the high pressure technique. That is the use of autoclaving. So most of the equipment you are going to be using, or most of the laboratory wares, you should make sure that all these are free from contaminants. So you could autoclave them so as to prevent contamination from growing there. Okay, so you autoclave between 15 and 20 minutes. 15 and 20 minutes at a very high temperature of 121 degrees centigrade. So it is believed that at this temperature is above the temperature of water. So no microorganism, it is expected that only few microorganisms will be able to withstand this temperature. That's why that is the principle of autoclaving. So the principle of autoclaving is to sterilize by the use of high pressure for 15 to 20 minutes at 122 degree. So then another thing is to use dry heat. You can also use dry heat for our glasswares. We use dry heat for example, why do you have to use dry heat? So you have to make sure that you use dry heat. Apart from autoclaving, which is at high temperature, you can also use heat techniques that will make sure that you heat your tools, your laboratory tools, you heat them at very high temperature to prevent the growth of a microorganism. So that is that is number three. And apart from sterilization, the next thing is you have to maintain a very good hand washing techniques. Apart from the fact that you have used gloves, you should make sure that in between, by the time you are working, it's not going to be you have washed your hand in the morning and until you have finished in the evening before you now wash your hand. No, in between when you are working, you have to make sure that your hand washing techniques is excellent. You wash your hand, you disinfect it, the use of methylated spirit, the use of other antiseptic, wash with Dettol, hand washing. You will understand that hand washing, if you have a very good hand washing techniques, you are going to control a lot of infection or a lot of microorganisms. So don't forget that hand washing was part of what really prevented the spread of COVID-19. So to actually tell you that as a microbiologist, your principle of hand washing should be excellent. You wash with not ordinary soap, wash with detergent, wash with disinfectant. Make sure that your in between when you are washing is your hand washing principle is excellent. Okay, so it has to be before and even after. That is why you have to prevent They don't allow eating in the laboratory. So it's not that when you will be working, that is when you will remember that you need to go and eat. So by that, you forget to even wash your hands. And so that is part of the procedure to ensure that you work in an aseptic environment, aseptic condition, preventing. I told you aseptism or asepsis is a process or is a kind of a procedure that prevents, eliminates contamination or eradicates microorganism form of contaminants from your sample, so that whatever results you must we must come up with is not going to be that the your results you are actually working on contaminants. So every step of yours in handling microorganism, in handling your sample, must be such that is going to prevent contaminants from your work, from your environment. Make sure you do that. Okay, so I want you to also take time, like 1 or 2 minutes off, so that you'll be able to copy what is on the board. By the time we come back, we are going to talk on microscopy and staining. Microscopy and staining. Don't forget that as a microbiologist, it is not only your aseptic technique that you must know about. You need to know, you need to learn, you need to handle, you need to know the basic principle. How will microorganism appear under microscope? Then what are the techniques, what are the basic staining techniques you can use to make sure that they are feasible under the microscope? So that is very, very important. Okay, you are welcome back to the continuation of our educational series today, microbiology, with a focus on basic laboratory techniques in microbiology. Okay, before we go on a recess, we ensure that the first thing that you need to get as a microbiologist, the first thing that you need to score high excellently, need to score excellently in is just your aseptic techniques. You need to know more about aseptic technique. How can you keep the air around you, the environment around you, how can you keep it safe from contaminants or from another organism being contaminating it? So we look at that. We made mention of the first procedure that is very, very important for you is your PPE. It has to be excellent. After that, we also look at making sure that you work in a very sterile environment. So, and that can be through flame sterilization as well as the use of laminar flow, whereby the air around you should be well filtered. Then we also look at sterilization techniques of our tools and equipment, our media. So we look at the act of autoclaving, that is heating at a very high pressure, heating of your tools or equipment or media to 121 degrees centigrade for between 15 and 20 minutes. We also look at a must do procedure of dry heating your items, because you know that microorganisms cannot survive dry heat. So that's the reason why you also make use of high temperature in order to make sure that the air or the working environment around you is free from contaminants. So, and the use of personal protective equipment is very, very important, and this cannot be overemphasized. And that is when we look at gloves and laboratory coats and so on. Then we also said hand washing procedure, regular hand washing. You have to start washing. So the next one, our next focus is going to be on microscopy. So during our previous discussion, I told you about how microscope and the scientists or the microbiologists that discover or invented microscope. I call his name Anton van Leeuwenhoek. Okay, this Leeuwenhoek. So that is the father of microscope. So microscopy and staining will go together, because if you do not know the staining techniques under the microscope, you will not be able to see distinction between your field, the field of microscope, as well as what is coming from your sample. That was why the scientists now invented basic staining procedure. And the basic staining procedure that you should be familiar with, that at every point in time, is the one that has to do with the staining of the cell wall. So it is the cell wall that is going to be stained. So that means that's one of the reasons why you must learn gram staining techniques. Gram staining techniques that differentiated different types of bacteria into gram positive, I've mentioned that, and gram negative. Those ones. So under the microscope, when you apply the best staining or the most suitable staining techniques, the way each of the structure of microorganism is going to stain will vary. So the DNA is going to take up a different stain, the cell wall is going to take up a different stain, and you should be familiar with all this. So that is why let's start with microscopy. Uh, that is an act of magnifying your objects, magnifying your microorganism. Don't forget that we said microorganism. These are organisms that are too small to be seen with our naked eye. Forget it, you cannot see any microorganism with your naked eye unless you use aided eye, or unless you use magnifying lenses. It could be hand lens, it could be, and don't forget the size of bacteria. I told you that the size between 5 and 0.5 to 5 mm. That's the size. And for you to be able to see any, you cannot even see any bacteria without the use of microscope with these sizes. So that is why you need microorganism or magnifying objects to be able to see or study the microorganism. So there are two types of microscopy. The first one is called bright field microscopy. Bright field microscopy. This is the microscopy where light will pass through a specimen. And so that the shape, the sizes, and the arrangement is going to be visible. So what that means is that a bright field. So let's say this is your microorganism. So under that microorganism, if we are looking for this type of a microscopy, the light will be able to shine through, so that you'll be able to see the shape, the size, that you can even measure the size. The shape, is it going to be cocci, spherical in shape, or is it going to be bacilli, or is it going to be spiral, that is spirilla? I told you this in the last this. So, and that one also avail you when light shine. It's just like when somebody is in the dark. You are in a dark room, and suddenly light is shining into that room. You'll be able to see so many things. So that is what is called bright field microscopy. That's a technique that you make use of magnifying glasses or magnifying equipment, so that you'll be able to visualize the shape, the size, and the arrangement of your microorganism. Okay, and this is very, very ideal for dead. Bright field is very, very ideal for dead cells, that is dead microorganisms. So microorganisms that are no longer alive. So that you use bright field microscopy for that. So, and this is the usage. Now let's look at the limitation. That is, what are the challenges that you can come across when you are using bright field microscopy? The only challenge that you come across is that you are going to your specimen, you come across transparent, that you will not be able to differentiate your organism if it is not stained. So you are going to come across transparent specimen which are difficult to visualize. Visualize means to observe, visualize, to see without staining. So if you do not apply staining, That means your organism, your field is going to be transparent. So that is why bright field microscopy will only perform excellently, will only perform its function if you are using stain, suitable stain, to be able to differentiate between your organism and the field. Okay, the second type of microscopy is what is called phase contrast. Phase contrast microscopy. Phase contrast microscopy. Okay, this allows scientists like you, this allow you to visualize objects, to visualize your specimen without staining, without any staining at all. You don't need any stain to differentiate between that. So what that won't do is that under this, that's when you can see the nuclei, that is the nucleus, and that is when the vacuole. So if you want to see the nucleus of vacuole, that means you need to so if you are using ordinary bright field microscope to work, and you are telling me by the time you are not giving something to draw, that is, you are to draw whatever you see, and when you draw, you are not telling me that you are able to see vacuole as well as a nucleus under bright field. It is not possible. Bright field, what you will see under bright field will only be the size, the shapes, as well as the arrangement. Nothing more. Nothing more. So it is when you now get to phase contrast, and what is going to make you to be able to differentiate them, the size, the arrangement, is just because you will need stain to be able to visualize your specimen. But here, the second procedure of a microscopy, which is called phase contrast microscopy. So this will allow you to be able to see your specimen without any staining techniques, and under that, your nucleus will become so visible, and the vacuoles will also become so visible. Okay, so and the advantage of phase contrast microscopy over bright field is that this phase contrast will allow you to be able to, it is dynamic. It is very dynamic. If you say something is very dynamic, that means it is not stable, it is not stagnant, it changes. So you can vary it, you can manipulate it. So that is the dynamics or the dynamism of life processes. So you can still see. So don't forget that this one is meant for the dead cells, rather this one is one for what? To visualize living dynamic processes. So for example, if you want to study some metabolic activities like that will involve protein synthesis and some other, so at least under this you can easily be so sure that the life processes will still continue. Okay, so that is for that. So that is under microscopy. Now let me quickly mention something about the second aspect, which is staining. I've actually told you that stain can be categorized into two. You can categorize staining into two. The first one is called simple staining technique. So that is when you make use of a single dye like methylene, like crystal violet is applied. For example, now when you use iodine solution, when you use iodine dye, you are using only one technique, only one stain to be able to. So that means you must have learned that maybe your dye, let's say the color of your dye is like that methylene blue, it is blue. Now, you know that when you apply that dye, it is going to make it that anyone, so that means anyone that retain the primary color of blue, you will regard this as a positive for methylene. Anyone that takes up another color apart from the primary one, that means it's going to be negative for that. So if you are using methylene, for example, and don't forget that methylene is blue, so anything that apply blue under microscope, you will classify it as a methylene blue or methylene blue stain positive. It's going to be positive. So if it takes up another color like red, it could take up any color apart from this blue, it is going to be negative. And that one is very, very important. We make use of this, very, very important, in the diagnosis of some bacteria like rods, bacteria like rods, cocci. Very, very simple. So you can easily detect because you are using only one dye. Okay. The second type of dye or staining is, which you are very, very familiar with, is called gram staining. Gram staining techniques. So as a microbiologist, you need to know the procedure or the process of gram staining. Just like I told you that, and this one contains more than one dye. Unlike when you have a simple dye that you only have one, like methylene blue, you can have a hydrogen iodine, you can have any other one. But this one is a combination of basic dyes. Some dyes are called basic dyes. Some are acidic, okay, and some of them are both. That is, so you need to get to know that. So gram staining, we divide the organism into gram positive as well as a gram negative. So, and I told you that it will be positive, that is if the first dye you are going to make use of, the color is being maintained. It is the cell wall of the bacteria that this type of dye, gram staining, is going to stain with. So every stain will have its own target. There are some stains that their target is going to be only nucleus. There are some that is going to target the inclusion body, so that anytime you now see anything that is taking off that color, you'll be able to recognize that, this one is a vacuole, this one is a nucleus, this one is nothing but ribosome. So you need to know. So the gram staining, I told you that it contains more than one dyes. You have iodine. Apart from that, you have crystal violet. You have counter staining. So this one will go away that you will have a time you had dye one. The second one, you will counter stain. That is, you want to remove the first color. So whenever this first color is now being neutralized, so that means the organism can now take up a new color. But if the color is being retained, even if you apply counter stain, that color will still be maintained throughout, and that is when you will classify it as a gram positive. But if it does not retain the original color, that means it is going to be classified as gram negative. And I've told you that on the surface of a bacteria, we have the cell wall. The component of cell wall is not only that, it contains a very special component called peptidoglycan. So from the word peptide from protein, glycan that is from carbohydrates. That means peptidoglycan will contain carbohydrates and protein together. So, and I've actually talked about the thinness and the thickness of that kind of a cell wall. So I want to also give you time so that you'll be able to uh, use that time to be able to put one or two things down. Okay, I want to welcome you to the last part of this video. Just like I said, that this video is going to be like 3 or 4. The last part under these basic laboratory techniques that everybody, if you are going to be a microbiologist, you must get yourself familiar with. So the last part is a sample collection. How do you collect samples that is going to be of high quality? Sample collection and handling. Sample collection and how will you handle that sample so that you are not going to have a very bad result at the end of the day. Okay, so the first one, basic one, is the collection process or collection procedure. So what you must know is that your collection tools, what can you use to collect samples? So is it just anyhow bottles? Some of you just go and take anyhow bottles, needles, and these ones. Okay, so any collection tool you are going to use, you must make sure that they are sterilized. Sterilized is the condition. Needles, you can use needles to collect if it is a liquid like blood, cerebrospinal fluid. You can use needles to collect that. Then swab, cotton swab, you can make use of that. Okay, especially if you are going to collect from human sample, and you can use cotton swab so as to accept, but you must make sure that they are sterilized. So that's the reason why before you collect any sample from human sample, you need to get what is called ethical permission, so that after collection of your samples, you are not going to you supposed to help the patient. It's not going to be that you are going to now introduce contaminants, and it's not going to be microorganism that is going to cause, that is the cause of that infection that you are going to live there. You are going to, so you need to be very so we can also use wire loop to collect samples. Okay, use sterilized swabs, sterilized needles, sterilized other equipment to be able to collect your samples. You need that. Okay, now you must make sure that the tools you are using, I said, they are sterilized. Apart from being sterilized, they are not going to introduce external bacteria. That is, it's going to be bacteria. They are not going to introduce external bacteria. So for example, if you are supposed to get, sometimes contaminants can also be another bacteria. If you are supposed to get a sample that is going to you must see E. coli as being there. You're supposed to isolate E. coli, and by the time you now introduce contaminant, you are introducing contaminant, and you are finding any other bacteria that is not supposed to be there, but the bacteria is being introduced maybe as a result of you not having a sterilized container or collection this. So you can use loop, you can use sometimes you use bottles, like EDTA bottles, e.g. EDTA bottles. Sometimes when you are dealing with Mycobacterium tuberculosis, that is tuberculosis, so you can use sterile cups. There are specific sterile cups, but the only thing is that they must be sterile. They must be sterilized, well sterilized. It's not that it must have been broken from onset. So the first type of sample collection is It's going to depend on the tools you will use for collecting it. Then another thing is that it also depends on environmental um, factors, environmental samples. Okay, sorry. If you want to collect environmental samples, like maybe you want to collect from soil, what are those tools that you can use in collecting them? Okay, so you can use swab, you can also use swab like soil. You can use needles or other containers uh, that are very you are so sure of the sterility condition that is not going to introduce contaminants into it. So you need to know all this. It is very important. Now, I said sample collection. And one other thing that you need to know, time of collection is very important. The time you collect is very important. If you want to collect from a human sample now, for example, let's say you want to collect TB, that is sputum. Early in the morning, it is very best for you to collect early in the morning and late in the evening if you want to collect that sample. The reason is that whenever during the day, some of these microorganisms might not even be present in the required population. So that's the reason why there are specific timing of your collection is very important. That's why before you even dabble into collecting any sample, you need to know what you are looking for. For example, it's just like somebody who wants to study fish. Where will you go? You go to where there is a water. So you want to study fish and you are now looking for fish inside the soil. How possible is that? So you need to be so sure of the source. Where exactly can you target? Where exactly can you get your sample? It is very important. Then the timing is another very key part of that. So let's go to the last aspect, which has to do with handling of that samples. Handling of the samples will come under three important things. The first one is labeling. Any unlabeled sample can be confusing. If you don't label it, you label it, the time of collection, the site of collection, the type of sample. You also codename it. If you don't want to mention the person's name, you codename it, and so on and so forth. So that labeling is very important in order to prevent what is called a mixed up, so that the sample of Koinde will not be taken to be sample of Taiwo. And if one of them is being diagnosed with any disease, since you mislabeled them already, you are going to give a mixed up results, and you are going to make a very big error in terms of your prescription and so forth. So that's the reason why in handling, you should be sure of labeling. You should be perfect. There should not be any mixed up. You should label so that you have code and some. Then the last two aspects is the storage. You need to also know the storage condition of your sample. The storage condition at which your sample should perform best. For example, now there are some samples that you need to keep them under refrigeration. You need to keep them under refrigeration so that what you are looking for, especially those ones that heat, high temperature, increase in temperature, or instability in temperature can badly affect it, especially if you would do molecular analysis that is going to involve DNA analysis. You have to be very critical and very careful when it comes to that. So the last thing is timely processing. It's very important. So the population of microorganisms you found at hour 0 is going to be different from hour 8. It's going to be different from hour 16. It's going to be different from hour 32, and so on. So if you do not process your sample timely, you are going to have a very wrong diagnosis, or going to have a very wrong results at the end of the day. So all these are very, very critical. You need to be familiar with this. I've told you that these are the basic laboratory techniques that you as a microbiologist you must get used to. The first one is the aseptic technique. You need to be familiar. What do we mean by aseptic techniques? How can you make sure that aseptic technique is being maintained? Okay, we talk of a sterile environment, sterilization of tools and equipment and media. We talk of personal protective equipment. Then we also move to consider microscopy and staining, where we have microscopy, we have bright field microscopy, as well as phase contrast microscopy. The first one is being used for diagnosis of dead cells, whereas the phase contrast microscopy is used for diagnosis that has to do with dynamic life processes. So then we also look at staining techniques, simple staining techniques that will involve only one dye, and we also talk about gram staining that involve a lot of dye, including counter staining. And I told you that in terms of gram staining is a process that, or procedure that differentiates based on the thickness or thinness of the cell wall. I told you the major component of the cell wall is called peptidoglycan, peptide from protein, and glycan from carbohydrate. That is a mixture of protein and carbohydrate. And this will go a long way to be able to differentiate them in terms of their gram staining techniques. And this can be into gram positive and gram negative. And I told you about positive and negative. When you talk about positive, that means the sample or the organism in question is going to retain the primary color or the original color of your dye. And if you now have a different thing from that dye, that means it is going to be negative. Okay, so like gram positive, purple, as well as something bluish, depending on the organism that you use. Okay, gram negative will appear pink or red, whereas gram positive will appear purple. You need to know that. And the purple is for the crystal violet, which is the primary color. Whereas the red or the pink color is the color of saffronin, which is being used as a counter stain. So it is very difficult, it is very important for you to be able to differentiate based on their staining techniques. Then we also mentioned about sample collection, and that is the first one is a sample collection. How do you collect samples? What are the tools that you use in collecting samples? This is if you are collecting samples from very easy source like from human body, you can use this. And if you are collecting environmental samples like soil, like water, like any other, so you can also use some other containers in order to do that. And I mentioned that time is of essence. It's very, very critical. So the organism you have at hour 0 is going to be different from the population of organisms you are going to have at hour 32, and so on. That's the reason why you need to process them. Time is very, very important. Labeling to prevent mixture. Storage condition is very, very important. So don't forget that each of these topics or focus we are going to do, I'm going to send to you at least 16, between 16 and 20 objective questions that will be used to assess you. So when it is going to be, I'm going to communicate to you, but later on we are going to have the link that will be deposited on the below in the comment section of this video. Don't forget to like this video, subscribe, and also share this video. And don't forget to share the to hit the notification button so that anytime we now have any other lecture, you'll be notified. So I want to really allow for a break of more than 2 hours. We are coming back after 2 hours so that we'll be able to have the next lecture, which is going to be on cultivation and culture techniques. So that one is going to take up another video. Don't forget that I said this is going to be between 3 and 4 videos. So the second video is going to be a video that will be devoted for cultivation and culture or cultural techniques, where we are going to look at what do we mean by culture media, types of culture media, what are the factors that influence the type of media we are going to use. We are going to talk about shake plate method, pour plate method, selective and differential media, types of media, incubation condition, and so on and so forth. So don't forget, I will be waiting for you in the next video. Thank you.